Hello, and welcome to today's episode of ABCs of Neuroscience. Today, we are going to be learning about functional magnetic resonance imaging, more commonly known as fMRI. This brain imaging technique has become one of the most popular research methods in neuroscience due to its high degree of spatial resolution and minimal invasiveness. fMRI allows neuroscientists to get a look at the structure of the brain as well as patterns of activity in different regions, both during resting states and task performing states. To understand why fMRI technology is so popular, we first need to understand how it works. fMRI uses a rapid series of MRI scans to measure neural activity. By measuring the blood oxygen level dependent, or BOLD, response during this series of scans. You may be familiar with what an MRI machine looks like. But in case you're not, here it is on the slide. It's this large hollow tube that a patient lays inside. The tube administers a strong magnetic field to the brain and body that interacts with atoms and molecules in the brain. These interactions are then analyzed by computers to create representations of structure and function in the brain. In a regular MRI scan, the magnetic field emitted causes the protons and hydrogen atoms, which are in the lipid molecules that make up most of the brain, to align with the field. Then, low-level radio frequency waves are administered, and the protons spin out of alignment. Once the radio frequency current stops, the protons realign with the magnetic field, releasing their own radio frequency waves as they do this. By measuring where these waves of energy are coming from and the amount of energy released, MRI scans can reveal the relative density of lipid molecules in the brain and therefore create an image of its structure. fMRI technology measures how oxygenated blood activity causes disturbances in the magnetic field during a rapid series of MRI scans. To understand how this works, we must first understand the concept of neurovascular coupling, which is the mechanism responsible for facilitating all neural activity. Neural activity leads to an increase of oxygenated blood flow to active regions, facilitated through vasodilation, or the widening of blood vessels. This correlation between neural activity and blood flow is what's known as neurovascular coupling. Oxygenated blood is needed to facilitate neural activity because the brain requires an incredibly high amount of energy to function. The primary source of this energy is adenosine triphosphate, or ATP, which is produced through the process of aerobic respiration, in which oxygen molecules combine with glucose, producing carbon dioxide, water, and ATP. Oxygen is crucial for the final stage of this metabolic process, which is called chemiosmosis. Neurovascular coupling is the basis of fMRI technology because within hemoglobin, the protein in red blood cells that carries oxygen, oxygen is bound to an iron atom, which is highly magnetic. The magnetism of iron causes distortions in the magnetic field of the MRI. By tracking these distortions across a rapid series of MRI scans, fMRI produces three-dimensional images of brain activity based on the assumption that levels of brain activity correlate with those of oxygenated blood flow. fMRI is a very commonly used brain imaging technique in the field of neuroscience, particularly in research because it offers high spatial resolution but is non-invasive. Unlike CT scans and PET scans, it doesn't involve any radioactivity. One of the primary uses of fMRI in research is expanding our understanding of functional specialization, which describes how different regions in the brain are specialized for different functions. Task Task-based fMRIs are often used in research to identify which brain regions are involved in a certain cognitive function. As well, resting state fMRIs can be used to compare different populations. On the slide is of an fMRI scan showing how neural activity is localized in different regions during attention-related tasks, semantic-related tasks, and social cognition-related tasks. This demonstrates functional specialization. Despite its many uses, fMRI also has some limitations. One main limitation is its lack of temporal resolution, as its imaging of brain activity is restricted by the speed of blood flow, and therefore slightly delayed. Another major limitation is that fMRI is ultimately an indirect measure of brain activity. It does not directly measure the electrical activity of neurons, but correlating blood oxygen activity. fMRI can therefore only determine general activity levels in different regions based on this indirect measure, but it can't determine whether neuronal activity is inhibitory or excitatory, the direction of neuronal signaling, or specific neurotransmitter signaling. To end this lesson on fMRI, I'm going to briefly present two influential studies that showcase the value of this tool. The first study is a task-based study where participants were shown images of body parts or inanimate objects. In this study, fMRI scans displayed consistent activity in the right lateral occipitotemporal cortex when patients were viewing body parts specifically, but not other objects, leading to the conclusion that this region of the occipital cortex is specialized for the visual processing of body parts. Similar studies that followed have discovered other functionally specialized regions of the occipital cortex, such as the fusiform face area, which responds preferentially when viewing faces. This study and others that followed have greatly expanded our knowledge of late-stage visual processing. The second fMRI study that I will present studied changes in brain activity of patients in non-responsive vegetative states when they were instructed to imagine two different types of scenarios. These scenarios were 1, playing a game of tennis, or 2, walking through all the rooms in their house. The fMRI scans in this study showed that when patients imagined playing tennis, their supplementary motor area was activated. But when they imagined walking through their home, their parahippocampal gyrus was activated. These patterns of activation match those of healthy control patients, indicating that the localized brain activity was not random and confirming our pre-existing knowledge of functional specialization. This concludes our discussion on functional magnetic resonance imaging, or fMRI. As we've now learned, fMRI is a popular neuroscience research tool known for its non-invasiveness and high spatial resolution. It measures neural activity by detecting changes in blood oxygen levels during MRI scans. While fMRI has limitations in temporal resolution and indirect measurement of brain activity, it has helped identify specialized brain regions and their functions. Two influential fMRI studies highlighted the occipitotemporal cortex's specialization for body part processing and confirmed the functional specialization in non-responsive patients. Overall, fMRI provides valuable insights into brain function and cognition. Thank you for watching.